we're trying to do 3D imaging. The display stuff is really beyond us. If I could display some of the wonderful ways you guys have shown so far, it'd be great. But things like this are just plain old projections through our samples. So what I'm going to talk about is the acquisition of the data that we've got. And the title is a little misleading. There's hundreds of people at our institute, the CRG. I'm just going to talk about what we're doing in our lab because that's what's going to be relevant for the consortium. So just briefly, what are we interested in? Electron microscopy is really not suitable for the scales that we're looking at. And similar, whole human things like MRI are not it. There's nothing wrong with these techniques. They're wonderful if that's what you want. But what we're really focusing on is this sort of what we call mesoscopic scale of samples in this kind of scale. Sorry, samples in this kind of size. Well, sorry. What we would like to see would be cellular details throughout intact organs or organisms. It's not that we can always see that, but that's sort of the goal towards which we're working. And the techniques that we're developing, the tools are SPIM and OPT, which I will discuss today, mostly OPT because I think it's a little more closely related to what goes on here. A bit at the beginning, because you're probably not used to look at these sort of samples, everything I'm going to show you is on cleared samples, which are dead and fixed. The techniques can work on live samples as well, but the data I'm going to show is on these fixed samples, and you'll see why in the slide. This little thing here is a mouse embryo. It's embedded in a block of agarose, and here it's in a vial of water. We take that, replace the water with methanol, and the thing looks something like this. You can see it's even actually a little less transparent at this stage. But the nice thing you can do is replace that methanol with benzyl alcohol benzobenzoate, and your sample looks like this, or rather it doesn't look like anything. If you zoom in on it, again, there's the mouse embryo here. If you look really closely, you can see the eyes. The pigmentation isn't cleared, but all of the soft tissue basically is index matched to this embedding medium. So you can take some really nice images because, of course, the samples are very transparent. The downside is, of course, that they're very dead as well. You can't do dynamics in this kind of situation. So optical projection tomography is one technique we've been developing in the lab. At least conceptually, it's quite similar to X-ray CT. We have a source of light, a lamp, white lamp in this case. We have a sample that we put in front of our lamp. Unlike X-ray CT, we have nice optics that work at visible wavelengths, so we can put in some lenses to form an image on the screen. So we turn on the lamp, light goes through the system, and we get an image of our sample here. This schematic is just a wide field transmission microscope. The main difference is that we add a rotation stage to the thing so we can turn the sample around, image it from multiple directions, and collect 360 degrees worth of views of the sample. That in itself is kind of interesting. This is a view of a mouse spleen. I just invert the contrast because it helps with the explanation a bit. You've got your 360 degree views that we take of that, which is kind of nice for visualizing, but it's not what we really want. So we do a reconstruction. If we just take one plane there, you can consider the intensity amplitude along that line. And what we do is we back project that virtually in the computer. So sort of take that and stretch it back through. That by itself is completely useless, but we don't just have one view, we have views from many different angles. So we take those and we start combining together, and you can see that the more views we add in, it takes a little while to build up, but you actually end up getting quite a nice cross-section along that yellow line of a thin section of the sample. And of course, you can do that for all of the planes in the thing, and then you have a nice voxel data set where you can scan through. These are just different reconstructed slices moving through the spleen. This works in transmission. You can also do it in fluorescence, just replacing that lamp we had there with a UV lamp for exciting fluorescence, some filters so that we can get the excitation light onto the sample, and filters, again, for detecting the light coming out. We get a nice fluorescence image of our sample. We can rotate it again and do exactly the same sort of process of reconstruction. I guess the point here is that we can do both of these things, the transmission and the fluorescence contrast. We get the nice reconstructed data sets that are co-aligned to each other. So it gives us this multimodal type of imaging. Now, the reason we're doing this, the technique was developed in Edinburgh, and we've brought it to Barcelona with us, is because we want to study in the lab mouse limb development. It's a biology lab, not really an optics lab. Sort of a sideline. In limb development, in a space of about 36 hours, this is a mouse, you go from a small, more or less amorphous blob with no structure into it to this sort of thing that's starting to resemble an arm with a hand coming out. So we want to study how do you go from such a simple thing to a complex thing. You've got this sort of emergent properties of the biology. And there's a lot of things that are going on. Patterning, morphogenesis, differentiation, mechanics all have to go into this sort of thing. What we'd like to be able to do is model the growth of this limb bud in the computer. So ideally, we would start with an initial condition, that limb bud I had over here, which wasn't differentiated, and computationally model how it develops, how it grows in the computer. So we started an initial condition with a finite element model. We can sort of see how it will grow over time. And if we can do that and we can get it to work properly, we get the feeling we understand what's going on. To do that, we're going to need a lot of data. We need spatial quantification of things like gene expression patterns, growth rates, the actual shape of the limb. So that's why we're doing this sort of 3D imaging. This is just one part of what we need to collect, but the standard method for looking at gene expression is doing RNA in situs, which basically put a dark stain in the sample where the RNA for a given gene is expressed. So this is too small to see much, but you'll get the idea in a moment. This is a limb bud at a certain stage expressing, labeled for the gene HOXD9. What we'd like to see is this sort of thing over many time points at different developmental stages and for many different genes. And of course, these are just 2D images. What we want is the 3D. As I said, to get these nice images, we have to clear the samples so they're dead. So what we're doing here is actually looking at different embryos. These are supposed to be turning, but you get the idea. It's 3D data showing the different gene expressions. This one is expressed mostly here in the spine, here in a couple spots there. We build up a library of these structures at different points in time. So now this is zooming in on a limb bud. This is the transmission image. There was an in situ staining for HOXA11 in this case, which is this dark stain here. Just to invert the contrast again, because it's easier to show things. This is the transmission data. This is the autofluorescence of it, just coming out, what shows up in the fluorescence channel. And you can overlay them, so you're getting a nice idea of the registered data. That you can see where the gene is expressed, and you can see the morphology of the limb. These are the raw projections. These are, hopefully, slices through the 3D reconstructions of the voxel data set. And I have to hurry along. I think I will just skip this. We can use this to correct these holes here computationally, but I won't go into the details of that. So yeah, what we want to do for the modeling, we require the 4D gene expression patterns, which we can get from our transmission data sets. We want the morphology, which we can get from the autofluorescence, and it's nicer once we do this correction. And we also need a lot more that I haven't gone into, but to do the modeling, it's more than just the gene expression and morphology. And I will skip this one for time's sake. The other technique summary, I'll also skip this because of a bit of summary at the end. The other technique we're using is light sheet microscopy, SPIM. This only works in fluorescence. The idea is really simple. You have a light sheet, you have a imaging system. This is just an objective lens with a camera behind it. You focus on the light sheet. You put a sample in there. It's attached to a stage, so we can rotate it to look at it from different directions and scan it in 3 dimensions. And we have some kind of a chamber, so if it's live, we can have a nice medium there to keep it happy. The real advantage to this over OPT is that because we're taking just images of single plane at a time, we can do tiling. We can zoom in at high resolution and then take many tiles over the sample to get a really high resolution data set. This is a schematic of the setup, which again, I won't go into. And well, this is what it looks like. It's a benchtop system with various different components on it. This is a sample of the head of a mouse embryo that we scanned in the system. You can see the eyes here are in white. Those were done in absorption contrast with the OPT. And the rest, the colors are fluorescence, 2 orthogonal views of the thing. The nice thing about this is, like, as I said, with the SPIM, you can really zoom in at high resolution and look at the fine details of these nerves in the face of the mouse. And then zooming back out. So what the system delivers is basically this high resolution, co-registered multimodal data. These are, again, max projections in 2 dimensions, but it's all 3D data in the end. So just to summarize, the OPT, we get our 3D information from the back projection algorithm. Gives us both transmission and fluorescence, and those are sort of useful together, not just 2 separate things. The SPIM gives us, the light sheet is what gives us our 3D imaging capability, and the combination of the 2 gives us this sort of multimodal imaging that allows us to get the best parts of both of them. And in the end, we can get sort of micron scale resolution and millimeter size samples. That was about 4 or 5 millimeters top to bottom. So just the acknowledgements. I'm in James Sharp's lab at the CRG. Jürgen Meyer is a very good student who's defending his PhD in a couple of weeks, and he's done a lot of the actual work here. This mouse embryo came from our collaborator in Paris, and of course the funding. So that's it. I would take questions, but there's probably no time. So thanks for having me here.